Today we're going to try and answer the following questions. What are the events leading to an action potential? What is the threshold of an action potential? And what makes the neuron's action potential less likely? When we're talking about our inhibitory postsynaptic potential and excitatory postsynaptic potential, we should be able to distinguish these answers. So let's get with the slides, let's talk about the information, and then we'll revisit the questions at the end. So here we have our neuron. Okay, the direction travels from here down to the axon terminals, which passes it along. The threshold voltage of the action potential is one thing that the excitatory or inhibitory state of the action potential is based on. The ion concentration inside and outside of the cell is another, and the permeability of the chemical channels. How prone are the chemical channels to letting the necessary chemicals in and out of the cell? Postsynaptic neurons use a variety of receptors. They use G proteins and channels to communicate. Medications can alter the neurotransmitter activity that alter the neurotransmitter activity, can cure or help psychological or neurological diseases. A neuron, when stimulated, fires an electrical impulses that travel down its axon. So it's going all the way down here, and it's coming into the axon terminals. Each neuron has only one signal that travels at the same speed each time and has the same signal strength each time it's fired. So there's no variation. Okay, so let's look inside a neuron. In this diagram, you see the presynaptic neuron, the axon, which is over here. The presynaptic neuron is this area here. And the postsynaptic neuron, which is the green. They're highlighting it up here also. And the chemicals utilized in both inhibitory and excitatory postsynaptic potentials. You see that sodium is shown in blue, potassium is in the magenta color, calcium is yellow, and the acetylcholine is the black, the little black dots here. Okay, all of these have a role in both of these particular processes. So depolarization of the membrane starts with acetylcholine being released by the presynaptic neuron. Okay, you see this acetylcholine, it was the little black dots. You see them there. They're going to be released by the presynaptic neuron. The acetylcholine is going to bind to receptors in the postsynaptic membrane. So it's going to come down here and it's going to bind to this membrane. At this stage, chemical channels open, allowing sodium ions to diffuse into the cell, going this way, and potassium ions to diffuse out of the cell. Generally, more sodium ions will diffuse into the cell than potassium ions diffusing out. The gain of sodium ions inside the cell causes depolarization. So that's your excitatory postsynaptic potential. This has the potential for triggering an action potential. So depolarization is triggered by IPSP, which is your inhibitory postsynaptic potential. It can happen after the resting threshold and before the action potential threshold. So take mine, that was one of our questions at the beginning. What is the threshold of action potential? Well, it starts with a depolarization. It leads to an all or nothing start of action potential. There is no part way with it. It either is going to happen or it's not going to happen. Okay, so hyperpolarization of the membrane begins with the inhibitory presynaptic cell, okay, connecting to a dendrite. Now you see the dendrites out here. They're on the ends of the neurons and the over here. And then it fires an action potential. So notice it fires an action potential to start to get the chemical channels to open and to allow chloride ions to flow into the cell, to diffuse into the cell, and potassium ions to diffuse out of the cell. So just like with EPSP, ions are moving in and out of the cell. Hyperpolarization inhibits any action potential from occurring. So it's triggered by an action potential, but the inhibitory state stops any further action potentials from happening, at least for a period of time. So let's take a look back at our questions. So did we answer the question, what are the events leading to action potential? Depolarization is what triggers an action potential, but inhibitory postsynaptic potential causes the excitatory postsynaptic potential. What is the threshold of an action potential? Starting with depolarization, it leads to an all or nothing start of an action potential. And then what makes the neuron's action potential less likely? That's in the inhibitory when the chloride ions are flowing into the cell and they hyperpolarize the membrane. That's going to inhibit any action potential from happening. So we covered all three of the questions and we went through the slides. So I hope you enjoyed the presentation. Thank you.